Hello everyone, my name is Cushing. Today I'm going to talk about MolDesigner, which is a web UI interface to design molecule with the help of deep learning. So first, some backgrounds. We all know that in order for a drug to work, we want it not only have high binding affinity to a target protein, but also it needs to be good at various drug likeness properties, namely absorption, distribution, metabolism, excretion, and toxicity, and also solubility and the lipophilicities. So traditionally, a compound needs to do various wet lab experiments to obtain these kind of property scores. And the human biochemist would then, based on this score, to modify the compounds, to optimize the compound using the domain expert knowledge. And this process would reiterate till the compound is good. However, wet lab experiments are very time consuming, and recently deep learning models have shown really good success in predicting binding affinity and also various molecular properties. And additionally, they can predict the scores within seconds instead of days for wet lab experiments. So in this work, we provide an interface for drug developers to use the deep learning predictions as like a real-time feedback to modify the molecule structure and reiterate on this process until the models, deep learning models, start to show the compound candidate has reasonable measures and properties. Cool. So that's the basic idea, and let me introduce each component of the MolDesigner and how it can help drug developers. So MolDesigner consists of input interface and output interface. So for the input interface, you can first specify the target protein's amino acid sequence. So here I just use the SARS-CoV-2 COVID-19 3-cell protease target. And then there's a drawing panel, which you can draw the molecules. So for example, you can like draw from scratch, using like keys, or you can like specify the atoms here, or you can load it from the existing molecules. So for example, we since we use 3CL protease here, we're gonna try the drug ritonavir, which is shown experimentally to be highly binding to the 3CL protease. So we go to the drug bank and we can just download the mol file here. Just copy paste it and then just put it here. And see we have this drug molecules loaded. And we can also modify the molecules based on and the expert knowledge, like adding a bound, adding like some items and functional groups and etc. But for the sake of example, I will just use the ritonavir. And then we can specify the deep learning models. So we use dpurpose, which is our library to do drug target interaction prediction and also drug property prediction in like in less than 10 lines of code, and you can find the GitHub repository here. And currently, the MolDesigner supports 5 drug target interaction prediction models, and for ADMET prediction, we support 3 models, but this is for each task, and we support 17 pharmacokinetic indexes currently. So in the backend, we have like around 50 ADMET deep learning prediction models. So now we can just simply click submit. In less than 1 second, 0.27 seconds, the deep learning models predict the result. And we see that at first it converted into the canonical smiling strings, and then it using the affinity prediction DTI model, it outputs the binding affinity, and it's using the KD currently. And we see that it indeed predicts really high binding affinity, 25 nanometers. And in addition to the binding affinity, it also predicts 17 ADMD properties like solubility, lipo, and 3 absorption properties, 4 absorption properties, and 2 distribution properties, for 5 metabolism enzyme targets and 2 excretion and clinical toxicity. And we also see that we know that ritonavir binds to this target really high, and this is also corroborated by our model prediction, since 86 is a pretty high score. Now, drug developers can, based on these results, and then go back to modify the drug molecule based on the domain knowledge. So you can think our deep learning prediction serve as like a proxy for the wet lab results, but they are in the real time. So now, for example, we can go back and modify a functional group of ritonavir. So for example, I'll just remove this one, and then we can just resubmit it. And then also in less than 1 second, it predicts the result again. But now we see that the binding affinity is really high, which potentially means that the functional group is important for this molecule. And yeah, so hopefully that exemplifies what this MolDesigner is doing, and that's it. And we are going to present MolDesigner in the Europe's 2020 demonstration session, and hope to see you there. Thanks.